Tēnā koutou, tēnā koutou, tēnā koutou katoa. My name's Helen Nicholson. I'm the Deputy Vice-Chancellor, External Engagement, and it gives me my great pleasure to welcome you all to this inaugural professorial lecture. Tonight we're here to celebrate the promotion of Professor Greg Jones. And it's great to see many colleagues and friends of Greg's here, as well as members of the wider community. And I'd like to give a very special welcome to members of Greg's family, his wife Rachel, and their children Amy, Samuel and Lachlan. It's great to have you here. My role is just to set the scene, and then I'm going to hand over to Professor Brunton, who's going to provide you with some details. And I'm personally delighted to be part of this special occasion, as I've known Greg since he returned to Otago in 2000, which coincided with my arrival at Otago. In fact, my first memory of Greg is meeting him in the anatomy mortuary over an amputated limb. So we've moved on a bit from there. Promotion to professor at the University of Otago is not something that comes easily. But Greg has shown very well that he can meet the very high qualifications and standards that are expected. He has been promoted as a research professor. He's published over 1,000 scientific papers, and as an external assessor of his applications comments, the quality of Dr. Jones' publications are outstanding. Professor Jones' research interests focus on cardiovascular disease, and he's been described as the leading figure in promoting multidisciplinary international team science to answer complex questions related to abdominal aortic aneurysms. And another referee commented that he is a pillar in the aneurysm community, respected not only for his science, but his ability to bridge personal differences and smooth egos. He is not only a leader in science, but a leader in the broad sense. So I'm out to embarrass you tonight, Greg. I also need to add that while Greg is a research professor, he is also an active and enthusiastic teacher. From my previous roles of Head of Anatomy and Chair of the Health Science First Year Operational Committee, I can attest to his many achievements as a teacher. Secondly, he's also been willing to take on leadership roles in teaching, convening the cardiovascular module for the medical program, and more recently, convening Hubs 192, one of the very large health science first-year papers. This is very unusual for a research professor, and I don't know of any other research professor who convenes such a large and important paper to the university. Greg has also contributed not only to the university, but to the wider scientific community through his involvement with the Otago Medical Research Foundation and with ANZCART, the Australian and New Zealand Council for the Care of Animals in Research and Teaching. I'd like to now invite Professor Brunton to talk in more detail about Greg's achievements, but before I do, I would like to finish with a quote from one of his referees, which I think sums up Greg as a person. Greg is perceived as a very intelligent person, but at the same time, he is humble. He is the really nice guy you want to have a drink with and talk about anything. So congratulations, Greg.
Kia ora, tēnā tātou koutua. My name is Professor Paul Brunton. I'm the Pro Vice-Chancellor for Health Sciences, and I'd like to add my welcome to that of Professor Nicholson's to this event this evening. My role is to summarise Greg's career to date and tell you a little bit about his lecture. And he has had an amazing career, so to try and sum that up in a few minutes is no mean feat, but I will do my best. So Greg obtained his Bachelor of Science with a double major in Anatomy and Physiology from the University of Otago in 1989. Following on with a PhD, again in Anatomy and Surgery from the University of Otago in 1997. Post Followed then at the Malagan Medical Research Institute in Wellington, back here at the Dunedin School of Medicine, and then on to the Physiology Department at the University of Melbourne in 1999. He joined the staff here at the university as a lecturer in January of 2000, as Helen has mentioned, was promoted to be a senior lecturer in 2006, a research associate professor in 2010, and then on to be a research professor from 2018, as we know. He has had, as I'm sure we'll all agree, a very successful academic career with multiple publications, grants, and many distinctions too numerous to mention this evening. So without further ado, I'd like to call on Greg to present his lecture, which is entitled "A Picture Tells 1,000 Words, A Visual Journey in Cardiovascular Disease Research." Thank you.
Kia ora everyone. First of all, thank you very much for all of you that have come along tonight and share this night. It's a very special night for me. I guess it's a once-in-a-career type of event, and I hope I can show you some of why it's not a hassle for me to get up in the morning and come to work. I love my job. I would do it as a hobby if they didn't pay me, but shouldn't say that in front of management, should I? So I'm just going to... So I decided I would talk about my journey through cardiovascular research in a visual context, because I think I'm quite a visual learner, and I hope that through this lecture you'll get an impression of that, of why really seeing is believing in many ways has been a sort of a principle throughout my research career. Now, my research began working with Bill Stebbins in the early 90s. Now, Bill was the foundation professor of pathology at the Wellington School of Medicine, and he sadly passed away a number of years ago now. But this was what was written about Bill by Robert Olson for a eulogy that was written for him. Bill was an amazing guy. He was a real intellectual, very old school intellectual, in I guess the Cambridge-Oxford sort of model. But what made him stand out was that he questioned the existing dogma in science. And he took on something that was really quite challenging at the time. He was a guy that said that actually it's not just about lipid as the driver of vascular disease, that there's more to it. It's the way the blood flows through blood vessels. And he really took a lot of flack for that. It wasn't a popular thing to say in the 1970s and 80s when he started his work looking at the pathogenesis of atherosclerosis. That's the pathology that tends to block your blood vessels. But he was also a very visual guy. And as an anatomic pathologist, I remember when I first met him, pretty much the interview involved him sticking the picture very similar to this in front of me and saying, "What do you see?" And really put me on the spot, and just sat there and looked at me and said, "Tell me what you see." And so my reaction to that was, well, I'm not quite sure, but I'd hazard a guess, and I went on from there. And I think it was that I would hazard a guess that probably won him over, because he did actually take me on to his group. This is actually a cell that's sitting in an atherosclerotic plaque. Its nucleus is this bit up here. This is the cell body here, and all this grey stuff around the outside is the fact this cell doesn't like being in this environment, and it's stacked up all this protein around it like a cushion around the outside. So after joining Bill's group, in a relatively short period of time, I managed to produce my first paper, and it made the cover of the journal, which I was really chuffed about. It wasn't the picture I would have chosen out of the paper, but it was on the cover anyway. And I remember it really hooked me. I got this idea publishing is pretty cool, especially when they put your picture on the front of the journal. So this was a paper actually that derived from work that wasn't the main reason I was working with Bill. He actually was really great in that he would actually foster your curiosity. And so he said, actually, there's this really old technique that these German pathologists used, and if you want to give it a go in your spare time, it might lead to something. And actually it led to about 5 or 6 papers, just that spin-off technique. It's called the Halschen technique. It basically means a thin skin, and it involves removing the endothelial lining off blood vessels and looking at it in isolation. And I'll show you some pictures of that in a moment. But when most people think about the cardiovascular system, they think about the big stuff. They think about the heart, the big arteries and the big veins. And these are big pumps and pipes with thick walls. And many pathologists and researchers tend to focus on the structural component of the wall. But in actual fact, often forgotten about is the fact that this whole network of pumps and pipes is lined by a really thin layer of cells that we call endothelial cells. And these provide the non-stick surface of our entire cardiovascular system. If you didn't have these little thin cells, the blood would literally set in your blood vessels and you'd have black pudding throughout your whole cardiovascular system, which wouldn't be great. So these cells look a bit like this diagrammatic representation here. They're a bit like big garden pavers that sit on the surface of your blood vessels, separating the blood from the wall. And they're really, really important. And they look like this. This is one of these Hauschen preparations. This is where I've peeled the surface lining of endothelial cells off the wall of a blood vessel and laid it out on a microscope slide. So this is what the cells looked like in the intact blood vessel. The nuclei are the dark, smudgy things, and the black lines I've used silver to stain the edge of the cells. It's a really old technique, and I managed to get it to work and produce these lovely preparations. I think they look kind of pretty. And it's like a cobblestoned arrangement that the blood would see as it rushes through a blood vessel. Traditionally, we look at blood vessels by slicing through them, a process called histology. We take the tissue and we slice it like on a ham slicer, but a really thin slice that we can then lay on a glass slide, put on a microscope and look at the structure of a blood vessel. And so this is histology of an artery wall. The blood would be up here at the top, and this would be the exterior of the blood vessel here. And I've put it in black and white because when I first started working, this shows my age, everything was published in black and white. And if you wanted colour, if you could get a journal to do it, they would charge you thousands for it. So everything was in black and white. But anyway, this is what a normal healthy blood vessel looks like. Now, my subsequent work, working with people like Andre Van Rij and the surgical community, we get surgical biopsies from patients that look like this. This is the end-stage disease. And what we've got to try to do is work out how we got from the healthy blood vessel to the diseased blood vessel and what were the steps along the way. And all I want you to take home from these two pictures is they look really, really different. And working out the steps in between is really tricky. And what Bill Stebbins had done was he'd developed some animal-based models to develop, to look at the steps that intervene between the normal healthy blood vessel and the extremely diseased blood vessel. And by doing that, you could have a step like this where you can sort of see a transition now from the healthy blood vessel, and you can see that this sort of looks the same. We can still see this black line here, which is a layer of elastic protein like rubber. You can see it's nice and intact here. Here it's become broken, and the muscle in the wall of the artery has flooded out and started to thicken this layer out here. And then you can go to another stage here, and the dark stuff in the middle here is actually fat, cholesterol, that's accumulating at the boundary where those breaks have occurred. So by using these models, we can actually understand the sequential process for the pathology of atherosclerosis, which is really important. What's important in this histology is that the endothelial cells are really hard to see if we look at them this way. They are a really thin layer of cells when you look at them side on. And so the elastic tissue is the black line there. The endothelial cells are a little, it's really hard to even point out, a little grey line on the top here. So most histologists and pathologists miss these cells in histology because they're very hard to see. So what would be better would be to use that Hauschen preparation where you look at the cells on face on, as the blood would see it running through the tube. And that's what this preparation is here, one of these preparations where we have the endothelial cells as a blood cell would see it inside the tube looking at the wall. And I've peeled this off the wall of the blood vessel. I'm going to sort of do that, animate that now, lift it off, peel it away and put it to one side. And that's the endothelial cells to one side. But then I'm still left with the rest of the wall, which I can also look at. And that's what this other image is over to the side here. This is the elastic tissue that those cells rest on. And you can see in this blood vessel, that elastic tissue kind of looks ripped and torn. I'll animate it. The green stuff is the intact elastic tissue, this rubbery layer. And the lines in between are where it's split and torn, like perished rubber. And what I hope you can see is that the endothelial cells sitting on the top have changed depending whether they're sitting on intact elastic tissue or they're sitting in one of the crypts of those tears. So if I put the blue animation in there, those cells are sitting on the intact elastic tissue. They haven't really changed much. But the cells that have flooded into the gaps where it's split and torn, and it happens very quickly, have become, they're smaller, they've proliferated, and I can tell that because the nuclei have a darker staining and they've more tightly packed into those gaps. And this was I think, quite a nice observation. At the time we were doing this work, everyone thought that the endothelium was a kind of a sluggish cell, and it got torn apart when these breaks happened, and they were slow to go in there and fill in the gaps. And in the time it took to go in there and fill in these spaces, you would have other cells coming out of the blood and getting into the wall. This is one of these preparations at where a blood vessel is branching. So I've got this diagram here. It's obviously not a blood vessel, it's a piece of metal pipe, but I think it's quite nice to show you this area here is the equivalent of that preparation of cells that you're looking at there. And that weld line that I've indicated with the black arrow is the equivalent of that axis that I'm indicating there. So what we're seeing are the cells as they hit one of these points where the blood vessel divides into two. So this is blood vessel A off this side, and this is blood vessel B over this side. And the red line is that axis or the flow divider that separates those two branch vessels. And what I hope you can notice is that along that ridge of that flow divider, the cells have an interesting shape. They're kind of almost rounded, whereas the cells running into the blood vessels have an elongated shape. These cells reflect the flow of the blood over the top of them. If the blood's rushing along really quickly, they become elongated and long, spindle-shaped. If the blood's kind of rolling around, as happens at the ridge of this flow divider, they can't really decide what shape to have, so they become sort of rounded. And the other thing you might notice is these dark purple splodges that are along this ridge as well. These are white cells, blood cells that normally run around in the circulation, and for some reason they've decided to stick at this point to the endothelial cells, and they're going to try to get into the blood vessel wall. And we know that's an important part of the process of this disease, atherosclerosis. So this work was quite neat because it's showing you that there's some cells land. This one's landed right on top of this endothelial cell. This one's gone to the edge between two cells. And this one here, it kind of looks a bit sort of dumbbell shaped. It's actually in the process of squeezing between these cells. And when we did this work, it was really the first to show these kind of associations. So this is a much higher magnification image showing that process in more detail. And what I did, which was a little bit different, and it took the old Hauschen technique from the 1950s, is I combined it with immunohistochemistry, which is a way of actually staining for specific proteins. So the brown you're seeing now are actually adhesion molecules, proteins, specific proteins, which bind white cells that come from the circulation. And so you can see this little white cell with the asterisks here, sitting here with this dark staining around the edge here, and you need these adhesion molecules. If you don't have them, the cell would sort of hit the top and bounce off again. And the analogy I like to use, and my kids will know why I use this one, this is an F-14 Tomcat trying to land on an aircraft carrier. And these cells are running through blood vessels at up to 2 meters a second. So a tiny little cell shooting along at 2 meters a second. They need some way to grip onto the wall so they don't get washed off again. And they do that with these adhesion molecules. And this work showed these adhesion molecules, a bit like Velcro, grabbing this cell. It's an interaction between the endothelial cell and the white cell. And the adhesion molecules are acting like the catch wire for this Tomcat trying to land on the aircraft carrier, gripping it down so it can stick onto the surface of the endothelial cell. It then rolls to the edge and tries to get under the endothelial cell into the wall. And this image is some work I did with Bill showing exactly that, and I'll colour it for you so it's easier. The white cell is the red cell, the endothelial cells are the blue cells, and you can see that this cell has rolled to the edge between two endothelial cells and is burrowing down to get into the wall. So those adhesion molecules captured the cell, it rolls to the edge and then burrows into the wall. What does it do when it gets there? Well, it's going to start to change the underlying character of the wall and thicken it up and form the atherosclerotic plaque. But what this work showed, and this is some work I did when I came down to Otago with Andre Van Rij, was to actually show that these white cells aren't just arriving randomly at these sites of injury, that actually the endothelial cell is saying, hey, I'm the sort of incident controller here, there's an accident happened in the blood vessel wall, you guys need to come out of the blood and come in here and fix the problem. And you can see how that's happening here. There's a site of injury, the elastic tissue underneath here is broken. The cells of the endothelium have flooded in and actually repaired that pretty well. There's only a little bit here where the endothelial cells haven't quite covered up the defect yet. It happens really quickly. But the thing to notice here, the brown stain is a protein that these endothelial cells around the edge of this lesion, this defect, has started to express to say, hey, you white cells need to come in here, there's a problem. And you can see the way the white cells have responded. Previous to this sort of work, people thought they would land in the bit where there was no endothelial cells, where it was denuded. And it was this damage, the idea you had to damage the endothelium physically for the white cells to enter the wall. But this work shows quite nicely that the endothelial cell is actually the mediator here. It's the one producing the factors saying come in. And these are the white cells here, these dark dots here, here, and all over here. And you can see quite nicely that they're not landing in the site of injury, they're landing around the outside of that site. So it really is a very endothelial cell mediated process. And this is pretty important because we now understand that the endothelial cell is a key cell in atherosclerotic lesion formation. So those white cells land on the surface, they burrow through the endothelium, and then they get underneath the endothelium, and this histology shows that quite nicely. Here's the endothelium on the top here, a really thin layer. Here's a break in that elastic tissue, and you can see the cells have got in here and formed what we call a foam cell lesion at that site of injury. And that is dependent on the endothelium bringing those cells in. And this was a kind of, at the time, it's a long time ago now, but at the time, that was kind of a novel idea. And the important thing about this is it just keeps happening. So the cells keep coming in, and you can see here this longitudinal section of a blood vessel. You can see the blood travels from one end to the other and traveling through this, but it's really going to struggle because that plaque, that lesion, that diseased process has narrowed the lumen, the pipe, so it's harder for blood to get through. And if you look carefully, you'll see on this wall, there's a break here and here and here and here. This is where it would have all begun for this lesion because of these breaks and then this process of cells coming in out of the circulation to thicken the wall at that site. And the important thing that this is Bill Stebbins' idea about hemodynamics, the way blood flows in blood vessels is critical to where the plaques form. If we look at the opposite wall, that black line is perfectly intact. It hasn't broken, the endothelium's happy, nothing gets into the wall. And so we can see the lesion on one wall, quite a catastrophic change to the wall's character, the other wall completely unaffected, and that seems to be related to the way the blood runs into this vessel. It's hitting actually onto this wall where the plaque is present. And this is kind of fundamental work and really quite important in our understanding of the pathogenesis of vascular disease. So this diagram, does anyone know who the artist is that drew this? The backwards writings, the giveaway? Yeah, Da Vinci, exactly. This is Leonardo da Vinci's. I'm not going to compete with Leonardo, of course, but this is, I think, quite a beautiful picture. But actually, this is the reality of what he was trying to draw. Now, this is something that I created. Well, the body created, and I filled it full of plastic. This is a vascular corrosion cast. And this is, again, part of why I love my job. You get to do these sort of semi-creative things. This is a heart where I've injected plastic, liquid plastic, into the major artery coming off the left side of the heart, the aorta, pushed it back in, filling up the coronary arteries, let that plastic set, and then dissolved all the soft tissue away. And I think they're beautiful. I love them. And the students love them as well. We use them a lot in teaching. You don't get justice from it here in 2 dimensions. You can hold them up as a 3-dimensional cast, and we rotate them around in front of light boxes. Really good for trying to learn how to understand a coronary angiogram as you're moving on into your medical imaging. They're just, I think, just beautiful to look at. And this is another example here where I've coloured the different coronary arteries going to different parts of the heart, and I've let the resin go right into the small capillaries around the heart muscle. And this third example here is actually a person that had triple bypasses. So these things here are actually veins which have been grafted onto the patient's aorta and down beyond those blocked vessels to other parts of the heart to supply. And I love teaching with these because I think they're so beautiful, and the students seem to really enjoy them as well because visually they're very appealing, but they're also really nice to look at. And so this has become something that started out as a sort of a research tool. It's rather a teaching tool, but it's become a bit of a research tool. Just as a point of reference, in the anatomy collection now we've done hands, so hopefully you can all recognise a hand. And this is very useful again for understanding the blood supply to the hand. And you can see a digit here. Now all the pink or red stuff is where the blood would be. So that's plastic inside blood vessels. And you can see up here in the nail bed and here on the knuckle how dense that blood supply is. It's really impressive how dense this capillary network is that supplies the skin. In fact, I had to ring bark this finger so we could see the underlying structure. So I've dissected away this pink stuff here from the surface here. These are kidneys. And we use this in third year renal teaching, and they're really lovely to look at. And does anyone know what this is? Someone said it. It's an eye. The medical students often think it's a jellyfish. But anyway, I don't know why they think I'm teaching them about jellyfish. Okay, but I think that's beautiful as well. All of this is the blood vascular supply to these organs. This is one in the collection. This is a liver, and it's got multiple colours. I won't go through what the colours are for, but this was actually a preparation that we made in tribute to Professor Mark Stringer, a great clinical anatomist and clinician that was in the anatomy department for some years, and this is now in the collection, the so-called Stringer liver cast. But the great thing about this is we can take this material, so at a gross level you can see the lobes of the liver and the blood supply, but you can actually pull this material out, stick it under a scanning electron microscope, and this is what that material looks like when you look at high magnification. Looks a bit like brain coral, doesn't it, from the Great Barrier Reef? But this is actually the network of capillaries in the liver that are the exchange surfaces, where blood comes from our intestines and the nutrients are exchanged with the liver cells. And they come through this network of fine little capillaries and then out through the central vein here on the journey back towards the heart. And again, I just think it's beautiful to look at. This one here is sadly from a person that had to have their leg amputated for occlusive arterial disease, but they also incidentally had extensive varicose veins. And when we cast the skin of this person, there's a blue resin here, we cast this large serpentine varicose vein, which again I think is rather pretty to look at. And the white stuff over the top is actually quite interesting. It's the blood supply to the wall of the vein. So we think of veins as pipes, but they're actually tissue and they need their own blood supply. So we call this the vasa vasorum, the vessels of the vessel. So you can see this network of little vessels that actually supply this bigger vessel. And I think they're really quite pretty to look at too. But this area over here also shows something that looks a bit odd. And I first saw this and I was a bit confused as to what I was seeing. If we zoom up to higher magnification, it looks like little balls of glass wool. And if we zoom up again on those, you can see these little tangles of what looks like glass wool. When the resin it's all the same colour, but as it gets thinner and thinner, it looks lighter and lighter. And this little tangle here is actually the blood supply to an individual sweat gland. So it's just a different way of seeing things. I mean, if you've looked at skin histology, you might have seen a sweat gland. You hardly ever see the blood vessels because they're all collapsed. But actually, when you do these casts, it's a really neat way of looking at it, which is of academic curiosity and interest, but and pretty to look at. But anyway, I think they're kind of cool. You can also take that small cluster of resin and put it under a scanning electron microscope. And what I thought was really amazing about this was that it's such good quality, the resin that we use, that it actually pushes against the surface cells, and a bit like making a plaster cast of the cell surface, you can actually make out the individual cells. So if I bring up that Halcyon preparation again, that's what it looks like. I'll animate 3 cells so you can see the shape of them, sort of spindle shaped. But look in that resin, you can see the shape of the three individual cells, these long spindle shapes. Because the resin's pushed so hard up against the cells, it's actually forced into the little gaps between the cells, and the nuclei have created these little raised areas here and here, which I think's kind of cool. And if we look at that tube in the back there, the cells don't have this long spindle shape, they have this rounded shape, and that's because this one here is a small artery where the blood's flowing quickly and the cells become elongated, and this one at the back is a vein. This blood is moving more slowly and the cells have this more rounded shape to them. So we can even tell whether we're looking at arteries and veins by the shape of the endothelial cells which line this cast. Again, this is a cast where I've injected the resin. This is another case with someone who's had to have an amputation, and we recruit from these patients. We ask them whether they mind us examining their amputated limb, and many people agree to this, which is really great for our research, and it's very generous of them to do that. And I've injected here into the resin into a vein here, and you can see the resin as it's got into the vessels under the skin. And when we dissolve the soft tissue away, that's the matching image of that, and that area there matches what's present in the cast. And if you go up to high magnification and you look in these venous casts, these vein casts, you can actually see valves, because in the venous drainage supply of our cardiovascular system, there are valves that make sure the blood goes back towards the heart. And by looking at these valves very closely, or the impression they make in the cast, we can work out whether the valve is working properly or not, believe it or not, based on the diameter of the vessel and the length of the leaflet of the valve. So there's one here, another one here, another one here. And we can take a cast like this, and I had a really great summer student some years ago, and summer students are just fantastic, and I think they're a great bunch of students that come in and spend 10 weeks doing a research project. And this summer student, Jordan Vincent, actually took those casts that I'd prepared and produced these maps, where the blue arrows and the red arrows represent those valves that we were seeing in the cast. And he mapped whether they were actually working properly or not. And on the basis of this, we actually, after only 10 weeks of his work, we produced a really good paper which informed really fundamental stuff around how varicose veins form and what happens to valves and which valves need to fail for bits of the skin to have refluxing blood. So it was really quite an important observation. And again, it was nice that Jordan could come up with this nice system for mapping out those very complex casts. So a visual representation of what was something that was very complex. But as has been alluded to, a lot of my work is not in the venous space, but in the arterial space, and particularly looking at abdominal aortic aneurysm. Now, abdominal aortic aneurysm is the most common form of aneurysmal disease, and aneurysm is just a ballooning of a blood vessel to an abnormal size. The problem with aneurysms is that if they balloon up too much, they can burst, and typically fatal if that happens. Particularly outside of a hospital. You can see here in this image of a person, a CT scan, the heart is here. Here's the aorta running down here, and the white space is where the contrast is running in the blood. And when we come down into the abdomen, where the tummy is, so this is the person's belly button around here, you can see the sac has formed out the front here, where this has formed an aneurysm in this person. This is a transverse slice through the abdomen. And so we have, to orientate us, this is the spine here, and this is the muscular abdominal wall here, and this is this person's aorta. Now it should be that size, and you can see that in this person, clearly it's much larger than that. As a reference, when the aorta gets to 3 centimetres in diameter, that's when we somewhat arbitrarily, define it as an aneurysm. But you can see in this person it's at 6 centimetres, and at this point this person needs to have this aneurysm repaired. At 3 centimetres we tend to watch it because there's actually more risk with the surgery than there is with the small aneurysm bursting. But at 6 centimetres it needs to be dealt with. So a lot of our work has focused on how we can look at inherited factors, genetic factors, and environmental factors to look at cardiovascular risk, such as abdominal aortic aneurysm. So we all know about risk factors like high blood pressure, high levels of fat in the circulation. We've got some biomarkers that we can test diagnostically that tell us about some of that risk. But our work has been started out looking at genomic or inherited factors that link to risk factors and also risk of cardiovascular disease. But of course, we all know that there's many environmental risk factors for cardiovascular disease. And in AAA, the number one risk factor environmentally is smoking. But what many people don't realise is that our environment and our inherited genome actually interact to form what we call the epigenome, where our DNA is modified by the environment or the lifestyle that we live in for our life. And so these M's here are actually for methyl groups, chemical modification of the DNA, changing the way our DNA is read depending on certain environmental exposures, such as smoking, a diet, etc. And so a lot of our work is looking at these genetic markers and these epigenetic markers as risk factors for cardiovascular disease. So I've been lucky. My time in cardiovascular genetics has intersected with a massive growth in the field. So at the start of this millennium, there was the discovery, or the sequencing of the human genome, announced by Bill Clinton in the White House. And then a few years later, there was this development of all these genome-wide studies for different cardiovascular diseases and a whole range of disorders. But actually, when we look at 2005, this is a pictorial representation of what we robustly knew to be genetic association for common disorders. And what these represent are different chromosomes. So that's chromosome 1 through to chromosome 22. Here's the X chromosome. Here is the sad little Y chromosome. And what I'm going to do is animate this up to show you what's happened in our knowledge about robust, and there's a difference between what was done before this time, which hasn't really stood the test of time, and more robust genome-wide associations. So I'm just going to animate this up, and so as we move into 2006, all of a sudden, bang, we start to see these little pinpoints coming up right across the genome, right across all these chromosomes, and it's just gone on and on and on, across a huge range of disorders. And so people say we haven't discovered much since sequencing the human genome. That's nonsense. And in fact, if I bring that up to date now, 68,000 associations robustly associated with a range of traits. That's things like cancer risk, heart disease, it's things like high cholesterol, blood pressure, etc., right across the genome. So we've discovered a huge amount in a relatively short period of time. And it's important to reflect on that. 750 of these regions are associated with cardiovascular disease or strong cardiovascular disease risk factors. And this is going to be my one sort of boastful slide. It's not a Kiwi thing to do, but our group's been really fortunate. We've been involved with international consortia which have published into this space with some pretty high impact papers in Nature, Nature Genetics, the New England Journal of Medicine, and particularly in abdominal aortic aneurysm. Our group has been involved with all of the genome-wide association observations linked to AAA to date. And it culminated in this paper that was published last year, which really summarised the association between abdominal aortic aneurysm and our inherited genome. And I was proud to lead that international consortium. And it was a lot of work. It was 120 authors. Just getting the paper into the journal was a marathon task. But the goodwill of a really multinational consortium was essential, and it's really quite a triumph, I think, for my career to have seen this one over the post. So what do these sort of studies look like? This is a Manhattan plot. Why is it called a Manhattan plot? Because it's like the Manhattan skyline. So what are we seeing here? We're seeing along the bottom here, these are the different chromosomes. And each dot represents a genetic marker positioned in its chromosome and in its position along the chromosome. So this is basically starting at one end of the genome and going to the other along this way. And the y-axis here, the vertical axis, is the strength of the association with the particular disorder. And this is the genome-wide association for abdominal aortic aneurysm. And so we're looking for these skyscrapers standing up here above all this noise down here. Now, the remarkable thing about this plot is that each individual that we looked at had 5.3 million genetic markers characterised for that individual. There's over 20,000 individuals involved in this study. And that means that this plot is not 1,000 words, it's 1.6 billion tests summarised in this one plot. 1.6 billion, okay? We've got a study planned for the end of next year, which is going to the plot that results from it will summarise 4 trillion genetic tests on one plot. So a picture doesn't tell 1,000 words, it tells considerably more at times. But I think that's remarkable. I think these numbers, if you'd told me this when I started my career, that we would be doing this kind of work, I probably would have laughed at you. But it's quite remarkable. So you can take an area with one of these skyscrapers, and that's what happens when we zoom up on that. And you can see here, these are all individual genetic tests. This is the peak SNP here, the peak association. And when you drop down, this is the region of the genome, and we know that in this part of the genome, there's a gene called IL6R. Now, you don't need to know what IL6R is, other than to say that it's a gene that we know to be associated with inflammation of blood vessels, and we know that's really important in AAA. So this actually has identified something that makes biological sense. So we've looked across the entire genome and found something that makes sense. What a relief. So again, with this kind of work, we're dealing with huge numbers of tests now. We don't just test a blood sample for one thing, we test for thousands of things. So this is a colleague's data set. This is actually gene expression data, so 28,000 genes essentially. This is what we call a volcano plot. And again, the higher up in the vertical axis, the stronger the association. If we go to the one side of the red line, we're looking at genes that have increased gene expression, to the other side, decreased gene expression, and we draw a line where we say above this line we think it's significant, and you can see lots of genes which seem to have significant decreased expression associated with having an aneurysm, and a few genes which have increased gene expression. So that's a gene expression volcano plot. But this is where the visual representation becomes important. This is our epigenetic data set, using the same plotting technique, the volcano plot. We're looking at changes in DNA now that are influenced by our environment. And when I look at this, there's 28,000 markers for the gene expression. There's nearly 500,000 markers for our epigenetic association here. But what I look at, and I see this is where the visual thing comes in. I see something very different in the pattern. On this side, I see like a set of wings. On this side, I see as like a bird wings versus a set of butterfly wings, because I'm seeing this funny little sub-winglet down the bottom here and here. And the question becomes, what is that? Why is that different about this data set to the other? What is it about that pattern? Can we make any sense of that? And it turns out those little winglets at the bottom are really important, because they are, if I color code, those are the parts of this epigenetic landscape which are influenced by what we've inherited from our parents. So some parts of our genome, they're pretty much the same in everyone. Other parts will all be different in the room. Those ones at the bottom there have what we call SNPs. They're different, polymorphic between individuals. And those polymorphic sites are in those little winglets. And we can use that pattern to identify them really clearly, and I think it stands out quite nicely. And that's why I think some of these visual representations can be really informative. Incidentally, that cluster up there are changes that are driven by smoking. And there's been a dozen studies that have shown that exact same set of clusters associated with smoking, and we see them in this aneurysm group. And that makes sense because these aneurysm patients are really heavy smokers. And believe it or not, this is also one of these data sets, again about 500,000 tests, and we're looking at it in what we call principal component analysis. And I just think it looks kind of cool, but it's also a real data set. And it's looking at the relationship between all these markers and how well they correlate with each other. But it's pretty to look at, but it actually tells us something very interesting, because when we start to funnel this down and winnow this down to a set of markers, we end up with something that looks like this, which again is quite, well, I think very pretty. But actually this set of markers and the relationship between them tells you what blood cells are running around in the circulation of that person. And you can use this set of markers to predict how many of all the different types of blood cells an individual has without actually measuring them directly. It's kind of cool. And if you've got a sort of an arty eye, I think the plot itself is just nice to look at. But it's real data, and it is telling us something really informative about blood. And that actually turns out to be a very useful thing to know, to be able to take this set of markers and actually predict someone's blood cell composition, and we use that a lot in our research. This is what we call a heat map, and this is an important one. It's looking at coronary artery disease, the #1 killer in our society. Coronary artery disease, the blockage of the arteries that supply the heart muscle itself. This is a study where this plot in itself summarizes 1,050 individuals. That's along this way. So each of these columns represents an individual, and for each individual, we're looking at the values for 500 odd markers down this way here as rows. And what I hope you can see is that the people that don't have disease, the coronary artery disease-free controls, and the people that do have disease have different patterns. And so we're using these epigenetic markers to distinguish from people with disease and without disease. There's some in the middle here that we really can't distinguish between, but most of the cases have these markers that are coloured green because they're going down, and these ones are red because they're going up, and it's the opposite for the controls. So we can visually represent this very complex data set in a way that makes some sense to us. Finally, another, again with the theme of visual representation, and going back to Bill Stebbins' idea about really challenging ideas and not accepting everything at face value. This is our Otago Southland population that we've been looking at for many years for the prevalence of aneurysmal disease related to a lot of the genetic work that we've been doing. And this is typical of what you would see in most countries around the world, certainly Western countries, where the prevalence of disease, if we use the standard 30 millimetre diameter, if you're larger than 30 millimetres, we say you've got an aneurysm. If we use that in our Otago Southland population, 4.8% of men meet that criteria, and only half that, or less than half that, of women meet that criteria. And more importantly, in some ways, if we look at the trajectory of prevalence over age, we see that the men seem to get more disease as they get older compared to women. But there's a problem here, and it's shown in this picture here. Men and women aren't the same size, and yet we're using a size, 30 millimetres, which is based on the size that we consider to be an aneurysm in men. But we also apply that to women, and that's silly. Okay, women have a reference diameter of 17 millimetres, whereas this woman's husband has a reference diameter of 21 millimetres, 20% larger. So we shouldn't be using the same size. And actually, there's a very easy way to fix this. We just adjust these. We divide the aortic dimension by your body surface area, and we adjust for your body size, and it works very nicely. In men, when we do this, we get the same result, because that 30 millimetres was based on an average size male. And so the prevalence data in this plot, using this aortic size index, which is just aortic size divided by your body size, comes up exactly the same, 4.8%. That's exactly what we saw before. But when we do this in women, we see something very different. And I hope you can see now that it's gone from 2.1 to 4.1%. This is important because around the world, aneurysm screening programs have focused on men over 65, and the belief that this is a disease of men. What this study shows you is that is incorrect, that actually this is not a disease just of men. We've just been using the wrong threshold. And just last week, I'd like to show something that's right up to date, we published in the European Journal a study that showed that correcting for body surface area identifies the true prevalence of aneurysmal disease in women, and that the difference that have been reported widely internationally in the literature and has actually influenced clinical practice, because in the UK they only screen men over 65, that it's really an artefact of that male-derived absolute threshold, and that was a mistake. So to all the students in the room, the younger people in the room, and my colleagues, we always have to question stuff that we assume to be right, but you need to know that it's right. And this is an example where questioning that dogma actually could change the way we deliver health services. And I hope, for example, in New Zealand that we'll do that, because we know we have a problem with aneurysms and in Māori women. Māori women have prevalence of aneurysmal disease, which is the same as men when we use a male threshold. So they've probably got even worse disease, because when we adjust for body surface area, I think we'll find that there's a significant issue with Māori women, which also accounts for why they tend to have their disease younger and more severely. So I'd like to finish there and just thank again you for coming along today, and I hope you've got a sort of a snapshot of what I do and why I love what I do. I'd like to thank people in the room, Andre, my long-standing collaborator, Vicky Phillips, who's worked with ever since I've come back to Otago, the rest of the vascular surgeons here in Dunedin, the vascular diagnostics lab, have done a huge amount of the scanning work required here. Sean and Michael, my colleagues in cardiology, are great collaborators. Sally McCormick, Tony Merriman, this New Zealand Vascular Research Consortium, a network of clinicians throughout the country that have really bonded together in a common cause, and of course my international colleagues, which it's really the cornerstone of a lot of what I do now. But most importantly, I want to thank my family. And Lachlan's cringing because this is a very old photo of him when we went to Vietnam. And he's considerably taller now, but none of this is important without you guys. So thank you very much.
Kia ora, Greg. Mark Thompson-Fawcett's my name. I'm the Head of Department of Surgical Sciences, where Greg's a very valued member, and it's my role to give him a vote of thanks for his IPL oration that you've just heard. And you did deliver, Greg, on demonstrating with images how you've told the story of your research, and Greg's a very effective communicator, and this has been demonstrated by his talk, but also his success, how he's worked successfully with clinicians who often sort of operate in a different paradigm, and he's been very successful communicating with funders because he's had a long stream of success with the Health Research Council and good funding, which has been good for our department as well, but also communicating his research through publications and in presentations, and when he presents in our departmental seminars, I can understand it, and it's you've seen a lot of he's got into genetics, and that can be quite confusing to the amateur geneticist. But Greg always breaks it down, as you've seen, to make it clear. And I remember Greg was around just as I sort of came to the department in 1999, 2000, and I can remember Andre was desperate to get this young, great, clever guy back, and I think there were competing interests. But it was a good call, Andre, and it's been very successful. So thank you, Greg, for everything you've contributed to the department as well. As Helen outlined at the beginning, you're very collegial and you contribute in teaching in other areas as well and in service in the university, and you're a fantastic academic and university citizen. And you've also demonstrated great dexterity, and I just seeing all those things fly through there, realise it's very appropriate that you're in a department of surgery. But maybe we don't have skills quite as dexterous as yours. But you've transitioned through your research into genetics, from starting off with the more sort of tissue-related specimens. And you're a very humble guy too. It took him quite a bit of cajoling to put in an application to be promoted to professor, several years, but it all looked pretty obvious on paper that he could have done it a bit sooner. So thank you very much, Greg, for your IPL oration. And before the final parting duty, I've just got to remind you that you're all invited to some nice refreshments and wine and nibbles in the Union Common Room, not the usual site in the staff common room, because that's busy. So it's over towards the link, and there'll be, I think, people to direct you. So, Greg, I have a gift here to give you as a final thank you for your IPL oration.